Hey, so good afternoon and welcome to this EHDN Platform Meeting. My name is Patrick White. I'm the chair of the Executive Committee of the European Huntington's Disease Network. I'm very happy to be able to introduce you to this platform meeting, which we are conducting in collaboration with HDYO and EHA, our partners. And before I hand over to the moderator and my co-chair, Åsa Petersén, I wanted to thank everybody for joining us today in the audience, but also our panelists. We have a very knowledgeable lineup who will help us unpack the outcomes of the important UniQure trial. And I wanted to remind everybody that while we have very knowledgeable people on the panel, we don't know everything, and so it's important that we in our discussion don't become too speculative. With that in mind, I hand over to Åsa Petersén. Thank you, Åsa, for moderating this, and the stage is yours.
Thank you very much, Patrick, and welcome to everyone to this very interesting webinar we have ahead of us. So we're going to start by two presentations. First by Professor Anne Rosser from Cardiff University, also previous chair of the Executive Committee of EHDN. And then we're going to have a presentation by Professor Sarah Tabrizi from UCL Huntington Disease Center in London, who will talk us through the top-line results from UniQure. After that, we're going to move over to the panel discussion, where each one of the panel members will share their perspectives on the results, and then we will open up for questions from all of you. And so if you have questions, please put the question in the Q&A in this program. And so in the panel, besides Anne and Sarah, we may have Professor Liam Gray, depending on his surgery schedule, to join us. We'll have Dr. Patrick Waite from EHDN, as well as Bonn, to join the panel. We have Dr. Ignacio Munoz San Juan from Factor H Foundation. We have Jenna Heilman from HDYO, and we have Dina D'Souza from EHA, and also HD family representatives. Here, we like to show you the disclaimers of conflicts of interest from all the presenters and panel members. And with that, I will give the word to Professor Anne Rosser to start telling us about the study.
Thank you, Åsa. I'll just share my slides. So thank you very much. So I'm going to give a bit of background to the AMT 130 trial, and then Sarah will present the top-line results. So first of all, I just want to make it clear that although both of us will use some of the slides taken from the UniQure investor webinar, neither of us are presenting on behalf of the company in this webinar. So what is AMT 130 and how does it work? So AMT 130 is a gene therapy. It's a tiny piece of DNA that codes for a microRNA that's designed to lower the levels of Huntington protein. So it's a Huntington lowering treatment. And it lowers both mutant and wild-type Huntington, but importantly, it also lowers the highly toxic exon 1 fragment as well as the full-length Huntington protein. So the idea is that the DNA persists long-term in the brain cells, thereby acting as a mini factory to continuously produce the microRNA. So in other words, it's designed to be a one-off treatment for lowering Huntington. In order to get AMT 130 into the brain cells, it has to be packaged into a viral vector. And this is a modified AAV5 virus, which carries the DNA into the cells and has a predilection for entering brain cells over other cells. The virus is modified so it can no longer replicate, and the viral capsule is eventually cleared from the cells, leaving just the DNA which persists there. So the viral particles can't cross the blood-brain barrier, so they need to be delivered directly into the brain through a stereotactic neurosurgical procedure. And this was done in the trial in a highly controlled manner in a small number of sites in the US and in Europe, with the European sites being Cardiff and Warsaw, and the European recruiting sites being UCL, Cardiff and Warsaw. So the aim was to place the gene therapy into the striatum so that it filled the volume of the striatum, and it's anticipated that retrograde transport from the medium spiny neurons will mean that some of the therapeutic is also distributed to the cortex. So AMT-130 was delivered using the ClearPoint convection enhanced delivery system. This takes place in an MRI using an MRI compatible stereotactic frame so that the infusion can be monitored. And importantly, there was a cutoff volume of the chordate of 2 centimeters. And this is a video that's going to show you what an infusion looks like. And this is actually from a different gene therapy study, not the UniQure study, but the principles are the same. So the gene therapy is mixed with gadolinium, so to make it visible in the MRI. And you can see here are the cannulae going into the striatum. And this is showing you the gadolinium gradually filling the whole striatum. So this is pioneering neurosurgery, and at this relatively early stage of clinical development, these operations take some time. They take around 14 to 18 hours. And incidentally, I have seen some posts suggesting that the surgery takes 25 hours. This isn't true. I think there may have been one particularly long surgery of 20 hours, but most are in the 14 to 18 hour time frame. However, of course, 14 to 18 hours is still a long period of time for a patient to be under anaesthesia. So I think it's just worth highlighting that there's work ongoing to try and work out how to make this sort of surgery much shorter, and potentially eventually even so it could be done perhaps as a day case. And to this end, the EHDN Neurosurgical Task Force, under the leadership of Liam Gray and Romina Aram-Badden, has been working on challenges to the neurosurgical delivery of gene and cell therapies. And they've been doing this over the last 3 to 4 years. And in September, there was an in-person workshop in Venice with surgeons from across the US and Europe to discuss how to try and address these sorts of challenges. So I'm going to finish by outlining the AMT-130 trial design. So it's important to emphasize that this is an ongoing series of studies, some of which can now be reported jointly. The first 2 studies were a low dose and high dose versus placebo study in the US. And in fact, the high dose is approximately 10 times the potency of the low dose. And there was a similar study in Europe, but without the placebo arm, because this arm was not approved by the European regulators. The placebo arm was a partial burr hole in the skull, and patients were kept under anaesthesia for an equivalent amount of time to the treatment arm. The placebo participants were offered treatment after 12 months, and 4 were suitable and took this offer up. So patients were randomized to high or low dose. And also there are other studies ongoing. And these are a study of high and low dose with immunosuppression to try and reduce the risk of adverse reactions to the viral capsid, and a study to assess whether AMT-130 can safely be delivered to participants with a lower volume of chordate remaining. So many of these participants, as you can see from this, are not in the analysis that Sarah will be discussing. So this analysis includes only participants in the US and European studies who completed 3 years of follow-up. So what is the participant group included in the analysis? So as you can see from this slide, this amounted to 12 patients in the low dose and 12 in the high dose groups. And these results were compared to propensity matched patients from Enroll HD. And the propensity matching, really important to say that this was pre-specified and also agreed in advance with the FDA. So you can see the outcome measures listed here. The primary endpoint was the CU-HDRS. Secondary endpoints were the TFC, symbol digits, Stroop reading test, and the total motor score. And these were all measured in terms of change from baseline at 36 months. And the supporting endpoint was CSF neurofilament light chain. And again, change from baseline at 36 months. So at this point, I'll hand over to Sarah, who'll go through the top line results.
I've just put a request in to share. For some reason, it's not letting me share. Got it. Okay. Can you see my slides?
I think that, yes, they're there now.
Thank you, Anne. Thank you for inviting me to come and give you the top line results. I'm presenting exactly the same slides that were presented by Dr. Victor Sung at the Huntington Study Group conference earlier in October. So I'm going to give you the top line results. So all of us working in the Huntington's disease field are working together to find disease-modifying treatments for this terrible disorder. Anne has done a beautiful job outlining the AMT-130 gene therapy. It's a one-time administration. There's direct surgical delivery to the striatum with potential axonal transport to the cortex, suppressing highly toxic exon 1, full-length mutant Huntington, and wild-type Huntington. Anne has also given a lovely overview of the study update and the external comparator group. And I will be presenting data from cohort 1 and cohort 2 in the high and low dose that had reached 36 months. The Enroll HD database is the primary external comparator. This was pre-approved, and this was authorized because it was run like a clinical trial. The data has been given courtesy of CHDI, and many of us who run and are involved in Enroll, there is on-site, off-site data monitoring, and it is a concurrent study to the ongoing trial. So the clinical measures that are used in AMT-130, for those who are not regularly seeing patients, the disease progression in Huntington's disease can be measured using a number of validated tools. There is the Composite United Huntington's Rating Scale and the Total Functional Capacity Scale, both which were used as outcome measures for AMT-130. The CU-HDRS captures multiple domains of the disease. It includes the total motor score, the Stroop word reading, the symbol digit modality test, and functional abilities with the TFC. The TFC scale specifically assesses the decline in functional capacity, such as whether you're able to work, manage your finances, and perform daily self-care. In both measures, the scale scores decrease over time, capturing the combined worsening of motor, cognitive, and functional domains. This decline is closely linked to loss of independence and daily function. So there was a pre-specified pivotal analysis, and I'm going to explain this in easier terms, but the pivotal comparison of the AMT-130 versus natural history was conducted with a separate statistical analysis pan protocol that was aligned with, approved and submitted to the FDA in advance of the database lock for the 36-month data point. Anne has gone through these endpoints and the change from baseline at 3 years versus the Enroll-HD propensity score matched external control. So, what is propensity score matching? Propensity score matching is a method used in rare diseases and is an approved method for rare diseases where it may be difficult to have a placebo group. That's typically in advanced therapies like a gene therapy and also in rare diseases. So in clinical trials, we want to compare two groups. People who get the new drug, the investigational drug, and people who don't, who are the control group. In a randomized control trial, people are randomly assigned to these groups, which helps make the groups similar in terms of age, health status. But randomization or a long placebo sometimes is not possible. Anne mentioned that in Europe, the sham was not approved, and this is 3 years of follow-up with a very complex and lengthy neurosurgical procedure. And so sometimes randomization or a long placebo is not possible, particularly in invasive gene therapies with very small numbers and long follow-up time of several years. And that's where propensity score matching comes in. It's a statistical method that tries to mimic the randomization process by making the two groups as similar as possible based on their characteristics. As I mentioned, the Enroll-HD set was used and approved as the external comparator group for propensity score matching. It's an approved method for rare diseases, and several gene therapies and advanced therapies have been approved both in the US and Europe using propensity score matching, external comparator groups. This is the baseline demographics and disease characteristics across the groups. This is the AMT high dose 130 high dose. This is the PSM external control group. They were well matched for age, fairly well matched for gender. CAP score very well matched, DCL very closely matched for the baseline TFC, symbol digit, Stroop word, TMS. So the study met its composite UHDRS primary endpoint at 36 months. And here in the high dose, this is the disease worsening here on the y-axis going down. And at the high dose, the deterioration was 0.38. So there was still deterioration, but it was significantly less in these 12 people who'd reached high dose than the external comparator group. And this was statistically significant. Based on these data, there was a 75% slowing of the composite UHDRS. This is the line graph. Again, on the y-axis is disease worsening, and here is time 12, 24, and 36. And you can see already at 24 months, there had begun to be a separation of the two groups, but by 36 months, you can see they're really quite well separated. The key secondary endpoint was the total functional capacity. This had a reduction of 60% in the AMT 130 high dose group compared to the external comparator group. And importantly, in the Barreto et al. Generation HD1 paper, which was published by colleagues at CHDI and Roche, where they analyzed the placebo group in Generation HD1, the TFC, even after 24 months, was really resistant to placebo effects and was resistant to placebo effects throughout. What they also found was by month 17, the total motor score placebo effect had washed out. And by 2 years, the CUHDRS and cognitive measures, the placebo effects had washed out. This is the TFC time course. This is the external comparator group. Again, this is the high dose group, and you can see here the difference at 36 months. The data was looked to see if it prevented clinically meaningful worsening, and it was found to slow clinically meaningful worsening by more than 60%. And so this is the external comparator group, and there was a 68% reduction in clinically meaningful worsening on composite UHDRS and 62% on total functional capacity. These were the other secondary endpoints, and the AMT 130 high dose showed trends supportive of disease slowing across all the other clinical subdomains. And I think this is important because you want to be sure that the data all suggests improvement of the endpoints and that they are concordant. So the symbol digit, there was a reduction in progression by 88%, the Stroop word by 113%, and 59% on the total motor score. The low dose, which was 10 times lower dose, as Anne mentioned, showed variable trends in functional, motor, and cognitive endpoints, and the data was really suggestive of a dose-dependent effect with some potential benefit in the low dose, but much more clearly potentially beneficial in the high dose. The secondary endpoint, or a key exploratory supportive endpoint, is CSF neurofilament. Now, CSF neurofilament goes up with disease progression. And this is a graph from Felipe Rodriguez and Ed Wild's HDCSF paper. And it is neurofilament is a marker of neural degeneration and injury. And in early symptomatic patients, as was in this study, we would expect CSF neurofilament to go up roughly by about 10% a year. And that is graphically shown here. The CSF neurofilament at the 36 months was below the original participants' baseline at a group level. So the high dose was 8% below baseline and the low dose was 4.7% below baseline. It was below baseline at 24, 30, and 36 months, and by 3 years below the original baseline of the before the start of the study. Now that suggests that in the high dose group, neurofilament is down about 30 to 40% of what is expected. The adverse events you can see here, mostly relating to the surgery or the lumbar punctures, which were necessary to assess CSF. It remained generally well tolerated. There was a manageable safety profile. The majority of SAEs, which were drug-related, occurred within the first few weeks post-treatment, and all fully resolved with steroids or symptomatic treatment. And importantly, no SAEs since December 2022. So in summary, the high-dose participants and the high-dose AMT 130 met its primary and key secondary endpoints at 36 months, with favorable trends across additional clinical and supportive endpoints. I've shown you data suggesting a slowing of disease on the composite UHDRS compared to an external comparator, slowing of the disease progression on the TFC compared to an external comparator at 3 years, favorable trends across other clinical subdomains of the composite UHDRS, CSF neurofilament, a marker of neurodegeneration, below baseline, and the sensitivity analysis was undertaken, which was in keeping with the primary statistical analysis. Generally well tolerated with a manageable safety profile and no new treatment-related serious adverse events. I'd like to thank the clinical trial study sites, both in the US and the UK and Poland. I'd like to thank the steering committee, the neurosurgical planning committee, but most of all, we're hugely indebted to the patients and families who volunteered their time and their very brave participants who came forward for the neurosurgery for this trial. And thank you for listening.
Thank you very much to both Anne and Sarah for these very clear presentations. And now we're going to move into the second part of this webinar, which is the panel discussion. And we'll start by each one of the panel members to give their perspectives. And so I invite Sarah back to give your perspective on the results, please.
Thank you, Åsa. I think for the first time from the data we've seen so far, I believe we have proof of concept data which shows that Huntington's disease can be modified in early symptomatic individuals. So I think this is a really big step forward for the field. What we've seen so far in this data set is that in those 12 individuals who received the high dose, the clinical progression of their disease appeared significantly slowed compared to what is expected from natural history studies, and it was supported by objective molecular evidence, and the molecular evidence was cerebrospinal fluid neurofilament, which did not increase over time as would be expected in a natural history study. As I mentioned, CSF neurofilament is a marker of nerve damage, and when the measurement goes down, as it did, this suggests that nerve cells are potentially being saved. So together with the molecular and clinical evidence, i.e. the lowering of CSF neurofilament and the slowing of clinical progression, suggests that the AMT-130 in those 12 patients at high dose is modifying and slowing disease. There are a number of very important points that I just want to highlight. The news is important proof of concept data. It was only tested in a small number of Huntington's disease patients. So I would say this is a time for cautious optimism, but we've still got a lot to do. The study, as Anne mentioned, is not yet complete. Those 12 patients will reach 4 years next summer, and the other subjects at high dose will reach 3 years. Another cohort has been dosed, and a fourth cohort has been recruited and will soon be dosed. So soon, as this is an ongoing study, we will have 4 years of data on the original cohort and more data from the other cohorts and people who reach 3 years. So importantly, the study was based on the data I presented. It met its primary and secondary endpoints, which have to be pre-specified in advance. There's been a lot of discussion in the field about the external comparator group, and this is new for Huntington's disease, although it's a really well recognized in rare diseases with regulatory agencies. Now, I think moving forward as we develop more advanced therapies like gene therapy or cell replacement therapy in Huntington's disease, we will be discussing external comparator groups more frequently. I talked to you and explained what an external comparator group and mentioned it's a validated method to assess how a highly invasive gene therapy. It's not perfect. Absolutely, we all acknowledge that. But it's the best we have for this sort of study. And it's challenging to ask people to have a complex neurosurgery and be in the possibility of a placebo group for 3 or 4 years when their disease may progress. The study did have a placebo in the US called the sham, but it was for the first 12 months only, which was what the ethics approved. They approved 12 months. These individuals then had the option of crossing over, and this is consistent with current ethical thinking and follows standard regulatory guidelines. Of those individuals who first received the sham and then rolled over into the active group, 4 of the 10 completed only 1 year. And this was because the crossover was not open at the time, as the FDA had not yet allowed the crossover, as there was not enough safety data from the active treatment groups. And those subjects were lost to follow-up. Of the 6 individuals who could roll over, 2 had progressed over the interval of 1 year, and their striatum volumes had become too small to be eligible. But 4 went on to receive treatment. 1 received the low dose and 3 received the high dose. However, the data from the sham was small, short, and has not been so helpful in those individuals when it comes to assessing 3 years of data. So I think the particularly positive aspect of this study is that there are 3 years of longitudinal data. And to my knowledge, there hasn't been another interventional trial so far where 3 years of data has been collected and presented. The number of individuals in this study is small. It's 12 people at the high dose, 12 people at the low dose. But as I mentioned from the Generation HD1 placebo group paper, the TFC measurement is resistant to placebo effects throughout the course of the disease. I mentioned the cognitive total motor score and CUHDRS do have placebo effects, but they're washed out typically by the end of year 2. And that's from that data set, the Generation HD1 placebo data set. But importantly, the TFC measurement is resistant to placebo. And I think this is why the TFC result is important here. So to me, the data suggests that lowering of mutant Huntington and all its toxic forms, including the shorter exon 1 fragment, potentially preserves nerve cells and in turn looks like in those subjects it's preserving neurological function or slowing decline in neurological function in the small number of individuals. So I think this is an exciting new development for the field, but we have a lot to do. The principal limitations of this study are that it was only a small number of individuals. Due to the nature of gene therapy, there is not a 3-year randomized placebo group, and we have to rely on external comparator data. The study is still ongoing, more people will be dosed, so we will get more data. But that's the nature of gene therapy. It's complex, involves small numbers of patients with long follow-up. Meanwhile, people are working hard on a scientific publication, so this data can all be presented in full. So my final summary is I think this is really good news for the HD field because for the first time, I think there's evidence in early symptomatic patients that the disease can be modified. But this is absolutely not the end. And I think we've got a lot to do. We have to build on these results. And to quote Winston Churchill, I would say this may be the beginning of the end, but not this. We're not at the end. This is the beginning of the end, I hope. We have a lot of work to do. And if we do this together as a community united, we will be stronger. And thank you.
Thank you very much for those comments, Sarah. And we're going to move over to Professor Anne Rosser from Cardiff University to share your perspective.
Thank you, Åsa and Sarah. So I agree with what Sarah said. I do think these are encouraging results, but of course, it really is, as you can see from the presentations, it really is early days. So I'm really, really keen that we see the continuation of this trial. It's really important that we see the data coming through from the patients who are already in the study and hopefully more participants coming through and longer periods of follow-up. I think one thing that I've been thinking about is, you know, AMT 130 and the extent to which it could be a future therapy. And, you know, currently, of course, as Sarah said, the surgery is quite complex and it's taking place really only in specialist centres. But I think it's very important that we don't assume that that is the only way that the treatment can ever be given. And I think obviously we need to know first that it absolutely does work and that the results are persistent, and so we need this data to come through. But I'm not a surgeon myself, but I've been part of the neurosurgical working group, and it's very clear that the neurosurgeons are very optimistic that in the future it would be possible to find ways of delivering AMT 130 in a much more efficient way. So, for example, to implant cannulae, which can then be, you know, can be done, everything can be done in an hour or so, and then patients can have the infusion even while whilst awake. So that's looking into the future. It's not present yet, and that would take some years to happen. But I think we shouldn't assume that 14 to 18 hours of surgery is the endpoint. That's what it takes for an early trial like this. And then finally, just to say that whatever happens to AMT 130, whether it turns out to be a future therapy or not, I think it doesn't put any of the other therapies out of the frame. In fact, I think it encourages us that Huntington lowering is looking very encouraging, and of course other modalities of treatment also really important, and almost certainly we'll eventually need a range of treatment options for patients.
Thank you very much, Anne. And then I'd like to invite Dina D'Souza to share her perspective. And Dina D'Souza is an HD family member and a patient advocate for many years. And she's been a board member of the EHA since 2016. And she's also the patient representative of the executive committee of the EHDN. So please, Dina.
Thank you for having me on this panel. As an HD patient, which I am doing very well, I have followed UniQure for quite a few years. I was first part of their ad board in 2018. So as with other pharmaceutical companies, I've watched things grow and thought at the time when I first heard the results, it's like, oh, wow, this is some future things that are happening. I was super excited when I heard the results. It is a science breakthrough without a doubt. Yeah, Sarah and Anne have touched on topics as such. It right now is a very invasive surgery. Doesn't mean that in a few years it will probably always be very invasive, but hopefully the technology will, or the concept behind the technology will change so that it will be more ready available for other people. But right now it is a future thing. But that doesn't take away from the results that have been presented. It is a small number of patients. No doubt, gene therapy has always and continues to be a very delicate subject and tricky subject. But I'm hopeful for future generations, not necessarily for me, but as also has been alluded, there's not going to be a one fix all for everybody. We're all individuals. We all fit outside of a box. So there is going to be a cocktail of things that should and will be available to us as patients. I know that there's been a lot of disappointment in the last few years, but I think we still need to remain hopeful in terms of what's coming. And this is definitely a life-changing result for the HD community. Still a long way down the road, still an invasive surgery. So there's a lot of parameters that still need to be tweaked out and redefined. And we as a community still need to be patient a little bit more. And we will hopefully get there one day.
Thank you so much, Dina. Really appreciate your comments. Thank you. So then I'd like to invite Jenna Heilman. And Jenna Heilman is the Executive Director of HDYO, which works to support and educate and empower young people, 35 and under, impacted by Huntington's disease from across the globe. Please, Jenna.
Thank you for having me. And Åsa, I'm always happy that you can pronounce my last name correctly. That's a rarity sometimes. I agree with everything being said, and I think the I really appreciated the HD Buzz article that came out in September in response with the analogy of the first domino. And I think that that's an important piece of it, because this is a key piece in the research puzzle that then will hopefully be trickled down to the community. I think overall as a community, because I don't want to restate what's already been said, I think that walking that fine line of cautious optimism and realistic expectations is a really important and ever-changing and challenging thing to do. And I would say that over the course of the past 7 weeks, that's become really, really important for us to continue to work on, because what we see is that there's such a need for therapies for all people impacted by Huntington's disease. I think with there being the first therapy, there's going to be a really important responsibility on everybody for that first process, because not everybody is going to be able to receive the therapy for lots of reasons. Could be location, could be cost, could be disease progression, all of those things. It's always going to be a challenge, and gene therapy, as discussed, presents itself different challenges that we all have to collectively work hard in order to properly educate the community on. But one thing too is that this community is impacted by generations. And I think there's the concept of being positive research news, which is really important because it doesn't just impact one study, that impacts so many other studies coming down the pipeline, whether it's better awareness by outside communities, better access to having regulatory conversation, proof of concept, whether it's Huntington lowering or the other mechanisms being studied, all of that matters. But families are also living with this disease in a variety of different ways every single day. So with having breakout research news, it makes people think, you know, I want to be happy and supportive for this, but my loved one just passed away, or my loved one is too progressed, or I didn't qualify for this clinical trial. Will I still be eligible for treatment? So it's very multifaceted, and that's where this continual partnership between scientific partners and the HD advocacy and community really matter to help mitigate some of those expectations, mitigate some of those feelings, providing that support afterwards. I mean, even the word treatment carries a huge weight. We saw when people use cure, that carries a huge weight. Now we're seeing treatment as a weight with investigatory treatment versus, or investigational treatment versus treatment in the clinic. Some people are really confused at that. So there's lessons learned with all of this. So yeah, I agree 100% that this is really exciting on the research platform. It has a lot of really important implications. I just want to share a little bit about what the overall global community goes through whenever they hear this information. And it's not to say that they're not excited. It's not to say that they're not appreciative, that they don't want a therapy. It's just a reality of what these families and community members go through every day. So we are very appreciative for the hard work on this and so many other therapeutic approaches that are being investigated. But I think it's important that we just understand that there needs to continue to be an emphasis on this partnership.
Thank you very much, Jenna, for sharing these important points, really illustrating how important it is that we communicate very well and how complex this whole thing is. Thank you so much. Then I'd like to invite Dr. Ignacio Munoz San Juan to give your perspective. And Ignacio is a Chief Executive Officer and Chairman of the Board of the Rumi Scientific, who has over 20 years of dedicated experience in neurodegenerative research and development. Not only that, he's also the president of the Factor H Foundation, which is a non-profit organization committed to improving the quality of life of patients suffering from HD in Latin America. Please.
Thanks, Åsa. And I just wanted to thank everybody at EHDN for including me, but most importantly for hosting this event. I echo what Jenna just said, that for many of us who are directly facing patients, maybe not in the clinic, but in their homes, the news created a lot of confusion for people. And I think, unfortunately, the mass media preceded a detailed scientific back and forth or clinical back and forth about the interpretation and the possible caveats to the results that have been reported by UniQure. These type of events that EHDN is now organizing, I think, goes a long way, bringing other professionals who are not familiar with clinical trials and the patient community to really listen to the news firsthand, rather than listening to the news through the BBC or some other media outlet. As a scientist, I would say that if I were to design trial results with 12 patients, that the data that was presented by UniQure on the clinical scales was probably as good as we would have ever imagined if we just thought about it. Now, as a scientist, we know that we don't have access to all of the information. It's a small number of subjects. There are criticisms coming from professionals in terms of the potential placebo effects in a invasive surgery. Data that is available but not publicly disclosed yet. I'm sure UniQure will do so at the right time, and I'm just hoping that both the professional and the patient community uses this time to come together, as Sarah said, rather than cause divisions, which I think it's something that all of us who are active in the field have begun to experience. And I think it's rather sad that what is potentially a transformative set of news, if indeed the results continue with more individuals and a longer period of time, is really life-changing in the context of lowering Huntington. The other thing I would say professionally is that we've never attempted a Huntington lowering therapy directly into the basal ganglia. And I think people forget that many of us were skeptic that this could be even done in symptomatic patients. And I think we need to take certain things as clear wins for the field. Gene therapy directly into the basal ganglia is possible, is well tolerated. A certain extent of Huntington lowering probably in the basal ganglia is well tolerated. None of these things were clear, certainly to me and many others, prior to the results of the study. And I would urge everybody to come together, be a little patient, but I think there's reason to be optimistic, including considering all of the caveats that people are highlighting in the chat right now in terms of distribution, placebo effects, the statistical plans, etc. But I think this has been one of the most important periods since I've been in the field, not just for the potential positive results, but also what it is highlighted about how the community at large needs to respond when potential positive news are delivered, as opposed to when potentially negative results are delivered. So let's keep this in mind, and let's continue to work together. And thanks so much for EHDN.
Well, thanks to you. Thanks for your very wise points. Appreciate it. And then we're going to move to Dr. Patrick Waite, who is the chair of the executive committee of the EHDN, and he's also a clinician scientist at the University Hospital Bonn in Germany. Please, Patrick.
Yeah, thank you. And of course, it's difficult to add insightful perspective here after we've heard from a broad range of very knowledgeable people already. I think what sticks to me the most is what Sarah said in the beginning, and then Nacho echoed, is that this is at best the end, perhaps the end of the beginning, as Churchill said, and there is a lot that we don't know yet, and we, as Nacho said, we need a little more patience. Our webinar here comes at a different time than the other webinars that we have seen earlier. There have been important new developments from the regulators. But the immediate learning we've had, in addition to all the science and the clinical outcomes, is what Dina and Jenna have highlighted, is how much it matters how this is communicated. Yeah, and we will always have to communicate this with incomplete knowledge and science, and that makes it, because there are lives involved and lives and livelihoods involved, this makes it very careful when we throw around terms like treatment, which can be interpreted in many ways. Yeah, so that's my comment on this.
Mm-hmm. Thank you very much, Patrick. Important points. And so with that, we're going to start taking some questions. And so an important question that has come up is, what is clinical meaningfulness? That's a really important question now when we try to interpret the data from trials. So I'll start by giving the word to Sarah.
Thank you. And thank you for everyone's really fantastic comments. So clinical meaningfulness, because this gene therapy trial has given us all and the community a very rapid having to learn about clinical trial validation and endpoints, and meaningfulness is one of them. So clinical meaningfulness is actually a measure that regulators use to describe or to decide whether a drug has a beneficial effect on things that matter for patients, how they think, how they feel, how they're managing their lives. So Christina Sampaio, who is currently the Chief Medical Officer of the CHDI Foundation, and he used to be a regulatory lead for neurology at the European Medicines Agency, knows a huge amount about minimal clinically important difference. And actually, Christina was the lead author in a paper that many people know from Hamilton et al. In Movement Disorders about clinical meaningful change in Huntington's disease. And they used a method in Parkinson's disease relating it to a quality of life measure, the SF-12. And I spoke to Christina recently about the clinical meaningful important difference and the paper. And Christina said that, I think, and I'm going to quote it verbatim because I think it's a very good description. So Christina said the concept of minimal clinical difference was originally developed for assessing the relevance of drug-related improvements. Later, it was adapted for changes in deterioration, but humans have a lot of difficulty in perceiving slow deterioration versus short-term improvement. Therefore, the values that are found in minimal clinically important difference are typically very large. And the only way to get out of this is to say the minimal clinical change in deterioration is, for the reasons explained, and because Huntington's is very slowly progressing, a crude method to assess the value of an intervention. So Professor Wild at UCL has actually published a very nice article recently on clinical meaningfulness, which I recommend you read. It's a big topic. It's a whole topic that would be a whole webinar in itself. But that article has been endorsed by several colleagues, and I recommend you read it. I hope that's helpful. It's complicated, but I hope that was helpful.
Mm-hmm. Well, thank you. Dina, would you like to comment? What would be clinical meaningful, do you think, for people with HD?
From a patient's perspective, being able to manage their finances for a few years longer, being able to go for walks longer, being able to dance with their grandchildren. I wear two hats, so I do know the science part of it, so I'm not going to comment on the science. I'm just commenting on the patient perspective. So all these little steps that would add quality of life to somebody with HD is clinical meaningfulness to me.
Thank you very much. Anne, would you like to comment on that as well?
Yes, I mean, I think Sarah and Dina together have done a very good job of describing this. And of course, I think, you know, one of the things that we're looking at here, as Sarah's said very clearly is we're looking at slowing decline. We're not looking at stopping decline. We're looking at slowing decline. So that makes it quite difficult to measure. And I think obviously in this study there was a quite clear slowing of decline. The longer one follows that, the more meaningful it becomes, the longer it persists. So, but I think Sarah's hit the nail on the head. You know, there's no very simple answer to what's meaningful for patients. And, you know, as Dina's very nicely said, even a slight slowing for a few years can make a very big difference when you have no therapy to slow the disease at all.
Thank you very much. Then we have a question about if it could be helpful to use a placebo group from another study for comparison.
I'm happy to.
Yes.
I think that's a very good question. I think if there are 3 years of placebo data from another study, I think that would be wonderful for that to be made available for the community. Roche has made the Generation HD1 2-year placebo data available to the community. And I think if there are 3 years of placebo data from another study, I think that adds to sharing data in the community. This is a time when it will help us all if we pull together, like Nacho said. This is not just for AMT 130, and Anne also said this is for Huntington lowering and therapeutics for HD. So we want to kind of work together. So if there is another placebo group with 3 years of data, that would be wonderful if that could be shared within the community and with UniQure.
Fantastic. Yeah. And I think, you know, we are such a community where sharing of that kind of data could be and hopefully will be possible. Okay. So we have questions about the cerebrospinal fluid mutant Huntington levels, because we haven't seen that data and people are wondering.
I mean, I'm happy to answer that as well. Yeah. I'm happy if you. I'm not sure why the CSF mutant Huntington has not been shown in recent presentations, but I can comment on the assay relating to this study. Many of you who work in this area, the CSF mutant Huntington assay is a technically very challenging assay. It's got low sensitivity. If you go earlier in the course of the disease, you can't measure it at all. And I mean, just slightly earlier, in, for example, in HDSS early stage 2, so it's not too far, much earlier than the subjects in this study. And there's only a very small amount of mutant Huntington in the CSF available to be measured because a lot of it is tied up in brain cells. So in this study, it is compounded by the fact that the striatum, very important in Huntington's disease, and I think Nacho really hit the nail on the head when one of the things that came out of this is really treating the striatum seems to have really made a difference. But the striatum is only 20 grams of 1,000 grams human brain, so it's quite a small structure. It's very important in Huntington's disease, but it's small in the whole size of the human brain. And I think this is where we have to go back to the preclinical work. In the original preclinical high-dose mini pig studies, which were published in Science Translational Medicine by the UniQure team a few years ago, they very clearly showed that at the high dose, they had very significant CS of mutant Huntington lowering in the striatum of the pigs, but they didn't see changes that were in the CSF mutant Huntington in the pigs. And I think that's because we don't know where CSF mutant Huntington is coming from, and the striatum is quite a small structure, and there's lack of sensitivity in the assay. So it might be in a gene therapy like this, and I think this is relevant for other programs, where a small structure is targeted, we may not see changes in CSF mutant Huntington. If we had the Huntington PET ligand, would be much easier. We'd be able to do a PET study, but we don't yet have that. So I think it's challenging here with the CSF mutant Huntington assay with just treating the striatum. So that's, I think, potentially some insights into, and just to finally finish, we know CSF mutant Huntington is important, but it's not a biomarker that's really useful for disease progression, because in generation HD1, in the high dose group at 8 weekly high exposure, there was very significant CSF mutant Huntington lowering, nearly 60%, and those subjects did worse. So there isn't a correlation with CSF mutant Huntington and clinical improvement. But I think that the answer is really in the preclinical data, in that I think the striatum is treated, and I'm not sure we're going to see a lot in the CSF mutant Huntington. But that's trying to explain maybe why the data hasn't been shown.
Thank you very much. Yeah. And so, of course, people are looking forward to learn more about the data that's been generated in this trial. Could you also comment anything about the imaging data?
I'm happy to answer that. I haven't seen the imaging data. This not yet been shared. It will be shared in the a future medical publication. Obviously, there was a neurosurgical procedure, a significant neurosurgical procedure, and those of us who were doing the study and running the study could see the effects of the neurosurgical procedure in MRIs post dosing. So I think that will affect the structure of the chordatum putamen in particular. We look forward to seeing the imaging data when it comes in a scientific paper.
Thank you very much. And so a number of questions have come in that are asking about the regulatory procedure. And so we don't have all of those answers, but may I ask Patrick Waite just to say anything about how it works with the regulatory procedures in general?
Yes, thank you, Åsa, for bringing up that question. So I'm a little bit involved with the EMA, but I can't speak for the EMA, which is the European regulator. But I can say that they are very aware of the challenges when dealing with rare diseases and very, let's say, specific interventions, and that these regulators know that, let's say, traditional placebo groups are not feasible for the reasons that we've heard in the introduction, which are the complexity of the intervention, be it gene therapy or neurosurgery, or like in this case, the combination of the two. Yeah. I think it is important for unconflicted players like EHDN and other groups to be in a dialogue with the regulators, who are, of course, under a lot of pressure also from, let's say, conflicted groups who are in the business of commercializing these interventions, to develop approaches like that. So what I can say is the regulators in Europe are open to this. I know that at the FDA, there are transitions and shifts going on, but it's very difficult to comment on this from Europe.
Thank you very much. And so questions to HDYO, Factor H, EHA. So this big news and this increased awareness of Huntington's disease that we've had during the last couple of months, have that led to more people contacting your organization, potentially more funding? How has that affected your organizations? We can start with Jenna.
I was just laughing at Nacho laughing to the funding point. I think that the number of questions that we're getting and responses are very, I mean, Åsa, you and I were at EHA when we got the news, and I was sharing there are 500 people that I know of who have my personal WhatsApp. chat capability after the news hit. So, and we were working around the clock to answer those kinds of questions. And I think that even looking at the Q&A, people are really wanting to understand how they can treat themselves, how they can treat loved ones. And so I think it's a lot of the questions have moved a lot towards those more logistical type questions of how does this impact me? So I think from that perspective, yes. As of late, I'm a part of a group called the Cures Collective, which is a group of neurodegenerative diseases in the US, thinking of multiple sclerosis, ALS, Parkinson's disease, where we're trying to work together more collaboratively on some big asks for larger scale awareness. So this news has, especially as of late, has really garnered some attention to HD, because these types of decisions that are made recently do impact other rare diseases and other neurodegenerative diseases. So, I mean, I think it's just always the challenge of working within the community and trying to filter those very personal needs. And I think that's been the majority of what we've seen are those personal one-on-one needs and support.
Thank you. Dina?
This has put HD on the map for sure. It's went public worldwide. It was, I think, on every news channel, which this is fantastic. This is all about raising awareness. So there's no doubt about that. But there's always a balance, and Jenna stated in her opening, balance for managing patient expectations. And that balance was, in this instance, very hard because treatment sounds to a lot of people like a cure, which is not, but even a treatment sounds like next year I could get this or the year after that. And there was a lot of people contacting the EHA, contacting HDYO, patient organizations. Where can I sign up for this? And it was a very difficult time. And I think there's always lessons to be learned about how to engage, or maybe how to, in the next roundup for news like this, how to deliver it to the patient population.
Thank you. Yeah, and Nacho, in Latin America, did a lot of people hear about it? And
well, not only Latin America, also in Europe. I mean, I think like most of people who work in associations, we got up, certainly in the US, I got up in the morning with tons of messages and WhatsApps and is it real? Including from professional colleagues who were getting phone calls from their patients because they had seen the news, and even the professionals were not prepared to answer questions because they didn't know this was coming. So I think that the main reflection for me would be whether there is a possibility of having a direct communication strategy between the companies, institutions like EHDN or HSG, who enabled the trials to happen in the first place, and the patient organizations, so that when something big is happening in a press release, there's been a prior conversation that something is coming, and how to deal with what it means for the daily lives of people affected with HD or people who are dealing with HD, because it was unfortunate that what would have been what could have been communicated in a way that didn't create so much confusion and angst really led to many hours of people's times trying to explain, even to other professionals, what the data meant. I don't think the community was prepared. And I think collectively, maybe we need to reflect on how to do things better in the future.
Yeah, and I think, I mean, we really are such a community, and I think this has also helped us to start communicating even better and to work together. We're quite an organized community, so we're fortunate in that sense. Anne, I was wondering, could you say something? There are people who, of course, would like to participate in trials like this, and how could people, are there possibilities to still join, or what can we say to people who would have liked to be part of this trial?
So I think in terms of this trial, I mean, it's not recruiting in Europe at the moment. That's the first thing. I mean, we very much would like to think that it will take place in Europe again. Future participants, and of course there are a lot of clinical trials that are open and coming down the line, and I think people need to think very carefully about clinical trials and talk to their physicians and get really good advice. And I think the best way to start, if people are interested in clinical trials, is to become part of the Enroll HD study. And I think in a lot of centres, many patients are recruited from Enroll HD, not only from Enroll HD, but it's a really good place to start.
Thank you. And so all these news have raised questions about genetic testing. Sarah, could you please comment on, does this change anything for pre-symptomatic genetic testing? Sarah, you muted.
Thank you. So this came up also at the HDSA webinar. I would say I don't think this changes predictive genetic testing. I don't think that these results mean that people should rush out to get predictive genetic testing unless they were already planning to do so. So predictive genetic testing is a very personal decision. And I would say that if a person had no prior plans to get predictive testing before these results, then these results should not change that. These are early results. We've discussed the meaning of the results, and I think they are an important step forward. But we don't have a regulatory approved treatment for Huntington's that really has profound disease-modifying effects. As and when and if in the future we get to that point, and we're all working very hard for that, it may change people's uptake of predictive genetic testing one day in the future if we can offer one or more different treatments. But for now, I wouldn't change any decisions about predictive genetic testing if there were no prior plans for it.
Very good, thank you. So very important points. Okay, so there is some questions about this virus. And can I ask you to comment a bit? People are asking, so if the virus doesn't replicate and spread in the brain, how does it reach other parts of the brain?
No, good question. So what's thought to happen is that, of course, neurons are connecting with other neurons, and some viruses tend to be transported down an axon and some backwards to the dendrites, and that's what happens with AMT-130. So it gets transported along the projections of the neuron, and that's how it spreads. So it's not replicating, it's just moving. And of course, what we don't know is how much it spreads in humans. So Sarah mentioned the animal studies previously, and the retrograde spread was seen in the primate studies. But at this stage, we can't absolutely know that that's happening or happening to the same extent in humans.
And there are even more questions about this microRNA. How long does it stay in the brain?
Yeah, so the DNA is the thing that should persist in the brain, and we've got no reason to think that it won't persist indefinitely. And the DNA codes for the RNA. So it keeps on producing the RNA. So the DNA, of course, may, it looks like it's been, it has retained activity over the 3 years at least that these patients have been followed up. And one would hope that it will continue to be expressed and be active, but only time will tell us just how long it will continue to be active in the brain. So we don't expect it to go away, and we expect it to stay active, but of course that can only be proven by following the patients up for long enough.
Nacho, would you have any comment to give to this discussion, given your all your experience in research and development?
Yeah, I mean, I kind of echo what both Sarah and Anne have said. I think in terms of the distribution of this virus, I think it's been fairly well studied both in primates and in the transgenic mini pig model of HD. That was critical for their decision to move the program forward. In those studies, the distribution through the terminals, both from the thalamus and deep layer cortical neurons, as well as through projections from the globus pallidus and the nigra. So it's not just targeting the striatum. If it behaves the same way in humans that it does in monkeys and in pigs, it's pretty much every major structure of the basal ganglia and the connected areas. And that may be very, very significant in the high dose versus low dose, because the difference is really related to the distribution of the virus, the way it's been administered. So that could explain the difference between the high dose results and the low dose results. In the mini pig study, they did see a significant effect on CSF mutant Huntington of about 20%, but it was much less than the deeper sort of parenchymal lowering of Huntington. So just to remind people that the expectation going forward into the clinic was that the effects on mutant Huntington in CSF in humans would be much smaller than what was observed with ASO therapies. So in that sense, I wasn't surprised that early on UniQure did not report very severe lowering of mutant Huntington. However, I do think it's important to show the data, because it does mean that you can distinguish a low dose versus a high dose. And, there's many unanswered questions that I think all the scientists want to know. And I'm sure UniQure will be forward with those studies. And if we take other gene therapies for other disorders, I think the Parkinson's field has shown that an AAV vector, which the DNA is episomal and is maintained in cells for at least, I think the longest study was more than 10 years after a single administration. So we have every reason to suspect that this will be a very long-lasting therapeutic.
Yeah, thank you. Yeah, so this is really, really, really promising. So unfortunately, we haven't been able to be joined by Dr. Liam Gray in Cardiff. And so I want to give the word to Anne to comment a bit on that. And also if you have anything to add about from the neurosurgery perspective.
So just to say that Liam Gray, I know, is actually doing a gene therapy case, not a UniQure one, but a gene therapy case as we speak. So he had been hoping to join us, but it's obviously gone on a little longer than he'd hoped. So I mean, I've said a bit about the neurosurgery already, and I think, the key point I'd like to get across on Liam's behalf, I'm sure he'd have had a lot more to say than I've been able to do today. But I think the key point really is to, this isn't the end game in terms of the results, but also the neurosurgery isn't the end game either. And as Sarah said earlier, there's a lot of work to do yet.
Thank you. And so we also have questions coming in. What can researchers in other rare disease fields learn from these developments so far? Can I give that word to Patrick to start commenting?
I mean, yeah, thank you. Huntington's is a paradigmatic monogenetic disease, and I'm certain that researchers from other fields are very, very closely watching what the developments here are. I think from a scientific point, we've already heard a lot on the ins and outs. I do think that also from a regulatory perspective, the other fields look at Huntington's disease, because the Huntington's community is for a rare disease very large, and also in a unique position because of its level of organization. And therefore, the hope is that advances that are made in the dialogue with the regulators will also benefit other disease groups.
And Jenna and Dina, from your perspectives?
I think fields from both sides, other diseases, we learn from other diseases learn from us. There is cross-sectional procedures. Gene therapy, as everybody's alluded, is a very complex procedure. And there is still a lot to be learned in this field. And whatever advances are in other diseases, we will look at that also and learn from that, and vice versa with this.
Mm-hmm. Thank you. Jenna?
Yeah, I mentioned it a little bit, but I think advocacy is a clear component where we can all lean on each other. In rare diseases, especially in Huntington's disease, I talk about this a lot. It's not like anybody will develop Huntington's disease who doesn't come from a family that carries the gene. You can talk about gray area and some things like that, but there's not as much advocacy effort from outside members of the community, because it's rare, so not a lot of people hear about it, but also there's no threat to their family members or friends spontaneously developing Huntington's disease. You know, thinking of Parkinson's disease, multiple sclerosis, ALS, some of those things, there's that fear out there. So if we can have some of these common things to work towards with advocacy about placebos and how to work together on natural history data and come together with those advocacy points with gene therapy, and for the US, clear pathways to accelerated approval and openness and communication and working together. I think that's where I see a big opportunity within different advocacy organizations working together, even across different disease areas.
Great, thank you. Yeah, so we still have questions coming in from all of you. We really appreciate it. So we have some more specific questions. And so this goes to Sarah. So what about the selection bias because of required minimal striatal volumes and screening failures due to insufficient striatal volume? Because the propensity matching was only done based on clinical characteristics, or was the striatal volume taken into consideration?
Yeah, thank you. That's an excellent question. The original propensity score matching was done on clinical characteristics, but for exactly the reason that this question is getting at, there was also a matching using propensity score weighting using the track HD and predict data sets. The cohorts were matched for striatal volumes. And when they were matched for striatal volumes, there was no difference. So there was matching on the striatal volumes. That data is available. It was actually shown at the HSG meeting in the Q&A session. And I believe that will also be included in publications. The track and predict data sets are historical data sets. Track finished in 2013 and predict finished around the same time. So regulators are not so keen on external comparator groups that are historical data sets. They want ones that are concurrent data sets, and they have to be run like a clinical trial, like Enroll. So there isn't imaging in Enroll, so that matching was done using track and predict data and then used to extrapolate. The data I'm sure will be shared in the any future publication, but it's a great question, very important that it was done for matching. And I think Anne mentioned the newer cohort 4, which is currently recruiting in the US, and they're looking to see if they can do away with that minimal striatal volume and do the neurosurgery with just on clinical characteristics. So that will be an important moving forward in terms of this treatment potentially being available.
Thank you very much. And so, yeah, so people are wondering, this paper, when can we expect it to be published and read it?
It's a good question. I believe UniQure are working on the publication with the steering committee and the neurosurgical planning committee. And so we would hope that that would be submitted soon, and that the data then will be available for the community, and that it will be shared.
Thank you. And to Anne, what can we expect would be the easiest way of reducing the time for neurosurgery? What is the group working on?
Okay, so I'm answering this as a non-neurosurgeon, just to make that clear. But so I think there are a number of different things that have been discussed, you know, different sorts of delivery devices, because different devices take different lengths of time to insert, and you know, depending on the device, the infusion times can be different. Other ways of speeding up the surgery further down the line are things like having indwelling catheters that are put into the inside in the brain. That sort of surgery can be done apparently within about an hour, or sometimes slightly less. And then, because there are no pain receptors in the brain, the patient can then be woken up, sit and read a book while, you know, the infusion is ongoing. So those sorts of approaches, I mean, I've just plucked out a few really, and I'm not saying that any of those are necessarily what we'll end up with, but they're potential ways of taking the surgery forward and critically getting the you know, anesthetic time down. So where we are at the moment is really because this is all incredibly new, and it has to be done in a really controlled way to, you know, make sure you're really carefully targeting the right structure with the right volumes, etc.
Thank you very much. So that sounds very promising. So with that, I want to thank all of you who are here listening to us and will listen afterwards, which is also going to be possible. Thanks so much for the expert participating here, all the people in the panel. Thank you so much for sharing your perspective and your expertise here with all of us. Also, thanks to EHDN and HDYO and EHA, which is supporting this event and which we're doing in collaboration. And we know there are a lot of questions still out there, some that there are no answers for at this moment, but we will collectively follow up with more information to all of you as soon as possible. So with that, thanks so much for participating. I wish you all a very good night and take care. Bye.
Thank you. Thank you.